So thanks for coming, everyone. For those of you who don't know me, I'm Justine Shaw, and I've been with CBCS for many years. One of the joyful aspects of being part of CBCS was getting to lead a research grant that I was funded for externally to work on Macquarie Island and to look at the ecosystem's recovery following on from eradication of rabbits, rats and mice. And that work also aligned really well and was complementary and covered as well under the NESP 4.2.3 project that I led, which was looking at eradication on islands. So I collaborated with Rich on this project and we advertised for a PhD student. We got lots and lots of applications, but one person wrote a very compelling tome of a letter that was highly piqued our imagination and started off with a very romantic natural historian sentence, and that was Jez. So Jez is going to talk today about the amazing work he's completed in his PhD, and I will stop talking. But Jez, well done on the amazing amount of field work, lab work and desktop work you've done, and from moving country to come and do a PhD.
Okay, thanks very much, Justine. Now you should have some slides appearing. Jaz, are they coming up?
Not yet, but sometimes there's a delay, so I'm just I can't see anything yet.
How's that? Oh, hang on. I might need to accept something. There we go. Share. How's that?
Working, Jez. We can see your blue screen.
Great. Okay. Yeah, so thank you. Good morning. Thanks, Justine. So, yeah, I'm just finishing my PhD at UQ, but I've been based for most of my time down in Tasmania. So I've not met many of you face to face. I'm delighted to be here in Brisbane in the flesh to chat to you all on Zoom today. Yeah, unfortunate timing. There we are. I just wanted to start by saying that I come from Kent in the southeast of England. There's a river that runs through Kent called the River Medway, and if you're born to the east of it, you're a man of Kent. And if you're born on the west of it, you're a Kentish man. And I was born on the east, so I'm a man of Kent. I also wanted to start just by acknowledging the many privileges that I've had through life, not least of which was the opportunity to foster a love of nature, which I did through sort of some formative summertime trips to islands around the UK. And particularly to their seabird colonies. And I was also extremely privileged to have this opportunity to spend time in Australia, and particularly to spend an incredible year down on Macquarie Island. The island's about 30 kilometres long with a research station right at one end of it. And the only transport's by foot. So you spend a lot of time out in the field, walking, climbing hills. It's cold, it's wet, it's very windy, but ultimately it's amazing. And you spend your days feeling like you're sat in a David Attenborough documentary. So yeah, it was a truly incredible experience. Why was I there? Well, to study petrels. Petrels are smaller cousins of the albatrosses. There are about 120 species globally, but they're rarely encountered because they breed on remote islands from which they can access marine resources. As a defense against avian predators like skuas, petrels dig nesting burrows underground, and they only return to these burrows after dark. So this also makes them incredibly difficult to study. Petrels are culturally important, so particularly through the Pacific, for centuries they've been harvested species, But today there are also tourist attractions with visitors coming to see birds returning to colonies after dark. In a natural state, petrels can occur in millions, consuming prey volumes that are sort of equivalent to other top order predators like penguins, and indeed sort of similar volumes to commercial fisheries. So Antarctic prawns, for example, consume 4 to 5 times more krill than the entire krill fishery each year. And in so doing, they transfer vast amounts of nutrients from oceans to island ecosystems where they leach into the coastal waters that surround them. And these nutrient fluxes provided by petrels drive terrestrial and coastal ecosystems. And the amount of nutrient being transferred, again, is sort of equivalent to amounts removed by commercial fisheries, or to nitrogen levels that are fixed by lightning around the world each year. So really significant inputs. But unfortunately, almost half of the petrels around the world are threatened with extinction. And by far the biggest reason for that is the impact of invasive species. While their burrow nesting habit is a good defense against natural predators, it's been no defense against mammalian predators. That was never a problem with petrels nesting on remote islands, but as humans have transported cats and rats and others to islands around the world, those species have gone on to have devastating impacts. And why were we looking at Macquarie Island? Well, Macquarie is a World Heritage Site and a Tasmanian State Nature Reserve. It's found about halfway between Antarctica and Australia. And sort of that top left map, you can see a ring of islands. And what's obvious is that the area of ocean is vastly greater than the small dots of land that are positioned through it. So for any species that have to return to land to breed, like seals and seabirds, these islands are incredibly important. Macquarie is floristically like the New Zealand subantarctic islands, but some of its wildlife is more related to other southern islands, things like king penguins only occurring further south where it's closer to the Antarctic convergence. And Macquarie is also unique geologically. It's one of the few places on Earth where the Earth's ocean crust has been thrust up above the sea. So it's an important and interesting place, but it's a heavily impacted place. It was discovered in 1810, and within 2 years, the entire population of 250,000 fur seals had been killed through hunting for their skins. People quickly moved on to elephant seals and to penguins, which were rendered down for oil. And their populations were heavily impacted in the process. But ultimately, more insidious was the threat from invasive species. And by 1900, 2 endemic land birds had become extinct, a native parakeet and a rail. And this sort of, the ongoing impacts from invasive species provided the impetus for pest management. Seabirds were the next worst affected after those native land birds, with petrels in particular declining rapidly when they first came to be studied in the 1960s and 70s. So as a result, wekas, which were a ground bird from New Zealand, originally introduced for food, were eradicated by 1988, cats by 2000, and finally rabbits, rats and mice by 2011. And while there was a huge amount of effort put into the eradication, there was no budget left over for sort of monitoring afterwards. And that's where Justine came in and secured funding for this project to take place. And a few of us looking at different aspects of recovery of the island ecosystem. While I want to share our research outputs today, I also sort of don't want to disguise the fact that this was my PhD project. And first and foremost, it's been a learning exercise for me. I've learned by doing, and I'm sure you'll be familiar with this, but I've found with hindsight, I do a number of things differently. And rather than meticulously planning a research agenda, I spent most of my first year writing permit and ethics and grant applications for the year ahead. So I sort of had this remit to go and see what was happening with petrels, and embarked on field work, spending time enjoying the wildlife, putting out sensors, acoustic sensors or camera traps to try to capture information about petrels, sleeping in converted water tanks, which were used as field huts, enjoying the amazing views, and spending a lot of time searching for burrows, searching at night with a spotlight for petrels, and even swimming out to some of the rock stacks around the island to search them as well. And after this amazing year of field work, it was then, you sort of step back and really ask what you've just done and why. And so I guess so that it quite neatly fitted into an overarching goal to understand petrel responses to invasive species eradication, but really looking at that through two lenses: the kind of stakeholder interest in threatened species recovery, but then our broader interest in the ecological drivers behind that recovery, and how the ecology of the island as a whole might be responding with increases or otherwise in petrel populations. And then it was a lot of sort of thinking and shoehorning that to get to that understanding of petrel responses to the eradication. I really sort of focused heavily on researching and developing survey methods that could give us reliable data, then using that data to actually examine the status and trends of species, and then taking a look forwards. So as you start to read papers about what people have done to survey petrels when studying petrels, basically everyone goes and tries to count all the petrels on an island. And that's what I'd done. And I sort of took a step back and tried to understand, well, why do we estimate petrel populations? And I guess despite being based in Tasmania, this is very much sort of UQ CBCS influence on asking the sort of important why questions. And it's sort of easy to see, this sort of desire for petrel populations or any population estimates pervades a lot of conservation, and it comes into things like this recovery plan. A clear example where we're asking for robust, up-to-date population estimates. But I also sort of had a sneaking suspicion from what I've been reading that we're not great at getting them. This is a very recent paper by way of one of any number of examples I could provide, where we've studied a petrel on an island, and come up with sort of confidence intervals of 500,000 to 900,000 burrows on the island, and then assuming that between 12 and 97% of those are occupied, we come up with a population estimate at the end of it. And to my mind, these aren't robust. And so what are we doing them for? What are we collecting them for? And I think an ideal way of doing this, again with the UQ hat on, would be a value of information assessment, where we'd get a load of managers in the room, and we'd ask them all sort of what they want a population estimate for, and then we'd work out, well, based on that, what level of certainty do we need to have around that estimate, and how much would it cost us to improve the level of certainty we have in the estimate, and is that worthwhile or not? But I didn't do that. I just did some thinking about it. And I thought that one of the reasons you might estimate a population is to collect a baseline with a view to repeating that in the future, and then comparing those two numbers and seeing if you can detect a change. And if you're going to do that, you need to have fairly high certainty to provide the statistical power that you need to be able to detect that change. Another thing you might do is simply to use your population estimate to feed into a conservation assessment. And there, if that's quite a small population that you're surveying, again, you want to have reasonably high certainty because it may affect what category of threat you end up listing that species as. And that might affect how many resources you put into its conservation. But if it's a larger population, purely from a conservation and assessment perspective, it doesn't really matter how much certainty you have in your estimate. If you want to assess the impacts of threats, for something like fisheries bycatch, you're trying to use your population data to see whether fisheries bycatch is having an impact. If it is, it might lead to fisheries closures, for example, and that has high sort of economic consequences. So again, you sort of want to have quite a high level of certainty in your estimate. So I sort of went through a whole bunch of reasons, and then I went through a whole bunch of papers and pulled out all of the published estimates that I could find. And I looked for the motivations listed within those papers for why they were collecting that estimate. And overwhelmingly, most people are doing it because they want to assess the status and trends of species. So the next thing we did was to pull out the level of uncertainty that authors are actually reporting in their studies around their population estimates. And we did a simulation where we simulated declines of 30%, 50%, and 80% across 3 generations, which equate to listing species as vulnerable or endangered or critically endangered under IUCN red list thresholds. And then we said, okay, well, based on the level of uncertainty in the studies that we've found, what proportion of those studies would be able to detect a change given those simulated declines after, say, 10 years or 20 years or 30 years? And we found that after 10 years, only about 5% of studies would be able to detect any change when a population is actually declining at 30% or equivalent to being listed as vulnerable. So it's not great. It seems that a lot of the estimates that are being collected and generated really aren't fit for that purpose that people have for them, which is to measure trends and detect change over time. And part of the reason for that is because petrels are super hard to study. So you need to find out how many burrows there are, but then you also need to know how many of them are occupied by breeding birds. And both of those things have uncertainty around them. And when you combine that uncertainty, it sort of blows out into your final estimate. So the main lessons that are coming out of this first part of my work were really to have realistic expectations. I think it's important that managers realize up front that they may not be able to get a population estimate that has sufficient certainty around it to deliver on the sort of aim, the reason why they're collecting that estimate in the first place. I also found that reporting of uncertainty, and particularly the underlying parameters, so the burrow numbers, the uncertainty around that, the occupancy and the uncertainty around that, is poor at the moment. And if we're going to improve on this stuff and really be able to pick apart what's going on, we need to improve on our reporting. We also found that we're better at estimating occupancy currently than we are estimating the number of burrows. So again, when you're going into designing your survey, that's something to be mindful of. We should be spending more time or more effort estimating numbers of burrows than occupancy. And we sort of considered whether new methods might help us in the future to reduce uncertainty. So I guess sort of by this point in my analysis, I was enlightened by this information and aware that the data that I'd just gone and collected might not be great for certain uses. So using it as a baseline for trend detection in the future, I had to be sort of honest with myself that possibly the uncertainty, whatever I did, was potentially going to sort of prevent that. So I focused heavily on other potential benefits coming out of the work, and one of those is looking at alternative methods to reduce uncertainty. I spent quite a lot of time looking at using camera traps to reduce uncertainty when estimating occupancy, but I'm not going to go into that work today. And instead I'll focus on the whole island surveys that I did to estimate burrow numbers. There are a whole suite of petrel species nesting on Macquarie Island, or thought to have nested historically. But I just focused on white-headed petrels and Antarctic prions, which both remained extant despite the impacts of invasive species. So they've persisted throughout. And I looked at blue petrels and grey petrels, two species which had been extirpated from the main island, but that have been recovering and recolonizing since invasive species eradication. And ideally, we would have a silver bullet of one survey method that would allow us to survey all species at the same time. So I wanted to look at whether unbiased survey designs, like randomized or stratified randomized surveys across the island, could help to survey and to yield data on all species at the same time. But we know from some previous work that those kinds of surveys can work pretty well for more widespread species, but can struggle when it comes to localized or rare species. And for those, people have used sort of biased, targeted survey designs in the past. The other aspect of this that I wanted to look at was comparing survey methods. So traditionally, people have used plots to survey things like petrels. You distribute a whole load of plots across the island, you visit them and count how many of your target thing are in the plot, and you extrapolate up to the wider area. And one of the challenges of that is surveying enough plots to minimize uncertainty. But the other is knowing whether you searched each of your plots thoroughly enough to detect everything that was there. So they're not good at dealing with uncertain detection. You make an assumption that you find everything. But an alternative to that is using distance sampling, which accounts for uncertain detection. By walking transects and measuring the distance to everything that you're finding, you can work out a detection function. And what that tells you is, as well as what you found, how much did you miss? So then you can use your raw data and adjust it based on how many things you think you missed to come up with a sort of total estimate. And both of these methods are really useful for giving population estimates, but they don't tell us much about distribution. And for managers, understanding the distribution of things can be really important for tailoring on-ground actions or informing monitoring designs, things like that. So I applied an extension of distance sampling, which is to use density surface models, where they take your distance sampled data and they model abundance as a function of environmental covariance. So this shows the relationship between Antarctic prion burrows and elevation in the top right, or vegetation cover in the bottom left, or slope. So I wanted to look at this and see if this could be applied to petrels. So a major component of my year was spent conducting these surveys. First of all, I set out on a preliminary survey covering the island to visit known sites and to understand what suitable habitat looks like for each of the species, and also recording burrows incidentally across the island when I came across them. I used information from a digital elevation model, layers like slope and aspect and elevation, to do a cluster analysis that identified strata across the island. So areas of similar environmental variables that could be grouped spatially. And I then overlaid all of those burrows that I'd found incidentally across the island on top of those strata, and looked for any habitat associations. So whether burrows were just occurring at random across the island in similar proportions as the strata that are present, or whether they seem to be grouped within particular strata. And we didn't find any blue petrels or grey petrels during that preliminary survey. So I only did this for Antarctic prions and white-headed petrels, where up in the top right there, you can see there were sort of clear preferences for particular strata. And previous work has shown that you're better off concentrating your survey effort in areas of higher abundance to minimize sort of variance around those areas. So we weighted our sampling design using the information from these different strata. So I assigned 200 plots across the island based on this weighting. So a third of the plots were positioned according to where Antarctic prions were found in the preliminary survey, a third based on where white-headed petrels were found, and a third were sort of control plots, just distributed at the same proportions as the strata themselves. So then I went off and surveyed them. I visited all 200 plots across the island and searched them thoroughly for burrows. And while walking between each of those plots, I distance sampled burrows along walking transects. That work, I think we came across 4 blue petrel burrows in total and no grey petrel burrows. So it was sort of obvious from the outset that it wasn't working for those localized species. So for them, we used a targeted survey design. So based upon the known sites, I had an impression of suitable habitat. I focused spotlighting searches, walking sections of the coastline at night to try to come across colonies. When birds are active over the colonies at nighttime, they're easier to spot, and you can detect them from further away than you can from a burrow in the daytime. So we did the spotlighting searches, and then I'd go back to any areas where I'd found birds at nighttime to search them by day and look for burrows. So from the weighted, stratified, randomized survey, we had 2 data sets, plots and transects, and I analyzed those in 3 ways. The plots, we simply extrapolated up based on the strata that they were found in, and the distance sample transects, we did a basic distance analysis, and we did density surface models. And broadly speaking, I found that all 3 methods delivered roughly similar numbers, sort of encouragingly similar numbers. Consistently, we found that distance sampling yielded slightly lower numbers than plots, and density surface models lower than distance sampling. White-headed petrels are relatively rare, though widespread across the island, and only a handful of the plots had white-headed petrel burrows in them. And we found about 300 burrows while distance sampling. And as a result, sort of overall uncertainty didn't differ massively between the different methods, and it was pretty high for either of the methods. Whereas Antarctic prions are more common, we found them in more of the plots, but not enough to sort of minimize uncertainty. Whereas we found over 3,000 Antarctic prion burrows during distance sampling transits. And that meant that the uncertainty around the estimates coming from distance sampling and from the density surface models was far lower. Using occupancy estimates that I'd sort of calculated using the camera trapping and other traditional methods, we were able to then adjust the burrow numbers by occupancy and come up with overall estimates and uncertainty around those for the populations of each species. And they were sort of reasonable in terms of uncertainty for Antarctic prions using the distance sampled data and the modeling approach. And searches were reasonable for blue petrels and grey petrels. But of course, they're not unbiased. We don't know what we missed through our biased survey approach. And lastly, I sort of wanted to show that we applied the same sort of modeling approach that we'd used from the distance sample transect data to the search survey data for blue petrels and grey petrels. So basically created a load of presences and a load of absences from all of the work that I'd been doing and used that to model distribution of abundance. And it massively over predicted for both species. So again, the sort of modeling approach just didn't seem to be appropriate. For localized species, you essentially can't create enough absences to really refine your model and to sort of constrain it. So as well as the estimates themselves, this work gave us an updated, a much better impression of distribution of these species across the island. The models predicted where Antarctic prions and white-headed petrels are, and our search surveys pulled out colonies, which I've circled because they're such tiny areas for blue petrels and grey petrels. And I just wanted to illustrate also what I was saying with the modelling. It just didn't work. It over-predicted for blue petrels and grey petrels. So the surveyed distributions are far lower than the model distributions. And I take some confidence in the survey distribution because we did all of this spotlighting and we covered a large part of the habitat that the models are suggesting is suitable and sort of know it to be empty. So ultimately we found there wasn't a kind of one-size-fits-all method for surveying this diverse assemblage. Distance sampling and density surface models were good for our widespread and most abundant species, Antarctic prions, but they didn't work for localized species. Ultimately, uncertainty is still fairly high if we were going to use this data for detecting trends in the future. So how can we measure change? Well, as is often the case with petrel surveys, unfortunately, lots of people say, oh, we couldn't compare our data with historic data because the methods differ. And this is far from ideal, but it's exactly the same case for our situation. The methods that were used to survey Antarctic prions and white-headed petrels in 1976 really weren't repeatable, and we just couldn't do the same thing, hence our different approach. But while the numbers themselves aren't directly comparable, there's no reason to suspect bias from either approach in the distribution assessments. So I have compared the distribution, and by scaling our data using the same thresholds that we used originally to scale Nigel Brothers' data, we're able to compare distributions then and now. And for Antarctic prions, there doesn't appear to have been much of a shift. They're sort of largely occurring where they used to. Whereas for white-headed petrels, there seems to have been a significant range contraction. And that sort of makes sense. We know that white-headed petrels had high adult mortality. They're being heavily predated in the 1970s. And their breeding success was very, very low. So we would expect that the population decline continued for the next 2 decades while cats were still present, up until when they were eradicated in 2000. So plenty of time for the population to contract. But the other thing we were able to do is to look at monitoring data from sort of constant effort monitoring sites, plots that have been established by Tasmania Parks and Wildlife Service and monitored a number of times over the years. And from that, we found that white-headed petrels and Antarctic prions, those 2 species that have remained extant throughout, they are now increasing, but slowly, whereas the 2 recolonizing species, blue petrels and grey petrels, are both increasing rapidly. When we look at the breeding success in the lower plot, again, it shows how breeding success sort of remained low up until cat eradication, when it shot up. And we think breeding success then declined as the rabbit population increased in the early 2000s, and it's since recovered for both blue and grey petrels, white-headed and grey petrels, since rats, rabbits and mice were eradicated. So all 4 populations are increasing, and importantly, and this is very much the stakeholder lens, looking at threatened species recovery, white-headed petrels and blue petrels no longer qualify as threatened under EPBC or state legislation, and we predict that grey petrels won't either in a few years' time. So this is, a massive success of the eradication, and something that we really want to broadcast to people now. So that's the kind of threatened species lens. But what's the ecological story behind this? What's driving these patterns, and what do we think is going to happen next? So petrels are seabirds. They worry about things at sea, but on land, to an extent, they basically need deepish, well-drained soils suitable for digging their nesting burrows in, ideally with good access to the sea and easy takeoffs. And beyond that, they're not necessarily massively fussy. The coastal slopes that you can see in the top left plot there that fringe the whole of Macquarie Island appear to be ideal habitat. But when we overlay the current distributions of all species on the island, you can see in plot D there that it's really dominated by Antarctic prions, and they are restricted largely to the interior plateau of the island. And these coastal slopes appear to be sort of underutilized. Why are we seeing the pattern that we're seeing today? Well, it's based on the ecology of these 4 different species. So blue petrels and Antarctic prions were formerly abundant on coastal slopes. But blue petrels have a more restricted climatic niche, and they don't occur at higher elevations. Antarctic prions have a broader niche, and they also occurred on the plateau. The coastal slopes were well vegetated, and as such, fairly protected for all species, but including all of the invasive species. So we think that weka and rat and cat populations were high in those areas, and that predation pressure therefore was the highest in those coastal slopes. And once eaten out of those coastal slopes, blue petrels had nowhere to go and they were extinct on the main island. And they only survived on some of the coastal rock stacks around the coast. Whereas Antarctic prions were able to persist on the plateau where it's more exposed and predator numbers were lower. Similarly, grey petrels are a winter breeding species, and so they faced higher predation pressure than white-headed petrels. And so when cats were food limited in winter, grey petrels were heavily predated and they also went extinct. So we think this suitable habitat's underutilized. This really is my favorite plot coming out of the whole project. What it's showing is habitat use by the 4 species today based on where they occur currently. We can see that the 2 species, Antarctic prions and white-headed petrels, that remained extant throughout are restricted to very similar habitats as one another. And the 2 recolonizing species are occurring in very similar habitats as one another, but that those habitats differ markedly between the recolonizing and the surviving species. So recolonizing species are favoring drier soils on steeper slopes further from ridgelines at low elevations. And that corresponds very much with the coastal slopes of the island. Whereas the 2 surviving species are found in wetter soils on shallower slopes further from ridgelines at higher elevation. And that really corresponds with sort of suboptimal habitat that is found up on the island's plateau. And so it seems these sort of surviving species are kind of tethered to this refugial habitat. And I think the reason for that, if you're a recolonizing species, by definition, you're immigrating to the island and you can choose to go anywhere, you're likely to choose the best bits. Whereas if you're a recruiting individual of a species that's already established on the island, the pull of an existing colony and philopatry, the sort of desire to go back and breed where you were born, is probably stronger than the push from those colonies being situated in suboptimal habitat. So it's a super interesting dynamic that we find at the moment. What does this mean going forwards? Well, as we said, sort of suitable habitat currently appears to be underutilized. I looked at that another way by taking the models that I'd originally done for looking at distribution and modeled presence-only data to look at potentially suitable habitat across the island and say, were these species to occupy all the areas that we think are that are equivalent to where they live currently, where will they be and how many will there be? And you can see for all 4 species, their potential to expand and to use much more of the island seems great for all species. I looked at this a second way for blue petrels, for which we were able to get data from other islands in their range. And those different islands have different invasive species histories. So we asked, if blue petrels are to start living on Macquarie Island at the kinds of densities and in the same habitats that they occupy elsewhere in the subantarctic, how many will there be and where will they be? And what's cool about this is that the sort of current distributions are nested within their potential distributions based on where they occur elsewhere. So both of these approaches result in estimates for these species that, rather than being in the thousands or tens of thousands currently, come out into hundreds of thousands or millions. While this approach doesn't take any marine processes into consideration, which clearly might impose constraints on ultimate carrying capacities, what we're able to do is to compare current populations with populations elsewhere in the subantarctic. And these numbers that we're coming up with seem plausible. There are equivalent order of magnitude estimates for each species from other islands elsewhere in their range. One of my key aims is to start to communicate this effectively. So while it's great that we think these species aren't threatened with extinction anymore, I want people to understand that we still seem to be a long way, both in terms of numbers and also time. It's likely to take decades at current population growth rates for numbers to increase to the levels that we see elsewhere. So to try and communicate that effectively, we are kind of at a frontier in invasive species eradications where we're going to have to start tackling larger and human-inhabited islands going forward. And to cross that frontier, I think we need to communicate really well our long-term visions for what ultimate recovery of these islands looks like. I've been really inspired by the use of dystopian and utopian visions and forecasting exercises elsewhere in sustainability science. So this is done commonly in relation to climate science. It's called cli-fi, where we kind of compare business as usual scenarios with optimistic visions to help people to understand what the future may look like. This hasn't really been applied in terms of invasive species in the past. But we have a different situation there. For Macquarie, the dystopian nightmares were really sort of underway already. So we have early reports of packs of wild dogs roaming the island in the 1800s, tens of thousands of cat-killed seabird carcasses littering the island each year by the 1900s, rabbits grazing the vegetation down into a moonscape in the early 2000s, and whole seabird colonies being washed away with soil erosion. And now, thanks to invasive species eradication, we're on a whole new pathway towards this brighter future. So I've taken these predictions and the models and turned them into a narrative vision of the island 50 years from now. And that's something we're looking to publish. And I'd like to share it with you now. So I'm just going to close by reading my vision for the future. Visitors and researchers alike will see the change long before they see the island. In the days at sea leading up to the first sighting of land, thousands of seabirds swirl around the ship. The white undersides of blue petrels and prions outnumber the spindrift cresting each wave. And rather than ones or twos, a constant stream of white-headed and soft-plumaged petrels arcs by. On the island, all must tread carefully, sticking to managed paths to save collapsing burrows in the super colonies that cover seaward-facing slopes. Now that these areas, once denuded by rabbit grazing, have revegetated, soil integrity has improved, and a superterranean labyrinth of tunnels through the undergrowth blurs the lines between the surface and the underground world. In places, the smell of ammonia is powerful, akin to the remnant penguin colonies around the coast, but more pervasive is the warm, musty smell associated with petrel plumage. By day, skuas lazily patrol the colonies, going from burrow entrance to entrance as the occupants sit silent within. As the sun sinks, a transformation begins. It's a rare privilege to witness it. With the full outcomes of invasive species eradication now realized, resources for biodiversity monitoring and conservation are focused where desperately needed elsewhere, and this may be the last time the petrels are resurveyed. A scan from land with binoculars shows the birds approaching en masse, loitering over coastal waters, waiting for the cover of darkness. At dusk, black silhouettes swarm like flies up and down the coastal hills. Revisiting one of the colonies established decades earlier when cats were eradicated, eyes must be given time to adjust to the dark. Switching on a head torch to write in a notebook invites a hail of petrel wings and bodies. Disorientated by the light, they are attracted like moths to a flame. Where once the night was silent, save for the wind, the slopes are bubbling with the purr and chatter of blue petrels, the aerial chases of white-headed petrels mapped acoustically across the black sky by their quick, quick, quick calls, and the mournful cries of soft-plumaged petrels, so forlorn when uttered without reply, have become an excited cacophony. There we go. Thank you very much. That's it.